You should have a few folders. This because in each folder you have an application of the Inca model. So you have some of them are for the River Lee, some others are for the Salmonsbrook. And also you have some folders that are called Inca N and others that are called Inca P. So Inca N refers to the nitrogen model and Inca P refers to the phosphorus model. We will do a tutorial with the Inca N model of the Salmonsbrook. So open the folder Inca N Salmonsbrook. Okay? So you have different files there. These are the files that are needed to run the Inca model. The first and most important file is the .par file, .par file. This is the parameter file. It is a text file. Actually, you can open it with a notepad. For example, if you right-click and do Open With and choose Notepad, you can actually see what's inside it. It's just number, but be aware that it's a text file that can be modified manually. Then we have another file which is called .dat file, which is the data file. It's where the input data is contained, so the hydrometeorological series of precipitation, temperature, hydrological effective rainfall and soil moisture deficit. You can also open it with a text file, and actually if you just drag it onto the notepad that you have opened before, you can see that it's also a text file, and it's formed by 4 columns. And each column is a time series of a variable. The first column is soil moisture deficit, the second is the hydrological effective rainfall, the third is the temperature, and the fourth is the total precipitation. So you don't need to modify it, but just be aware that you can visualize and you can import the values also using a different program, not just Inca. Then you have 2 files that are called .obs. So the files with the observations. In this case, we have observations of flow and nitrate concentration, the Salmonsbrook. These 2 variables are not needed to run the model, but are needed to compare the model results with what has really happened, so with the observation. And we use them for calibration and validation of the model. We have 2 files in this case because we have a file which is called sb_ea, which is the observations, the records collected by the Environment Agency, that's why EA. And the other one is sb_T21, which is the data collected by you, by times 21. You don't need to keep it separate depending on the source of information, but I wanted to keep it separate in order to have it a little bit more organized. You can also open it with a text file. If you do, you will see, for example, the first header, you will read reach 5 flow. So this is the flow referred to reach 5. Then we will see in the model what reach 5 is. You have the date and the value of the flow. So the first date is the 27th of March of 1956, and the flow was 0.65. So well, as easy as it looks. So this is the flow. In this case, I think you can easily modify the observation file, because if you have a new sample, a new observation that you collect, you can just go at the corresponding reach, and add it at the end of the time series. So if you collect tomorrow a sample and you have a new value of nitrate, you will write 17th of August 2016, and the value separated by a tab. Okay, And then the last file is the .ssf file, which is the structure file. It is also a text file, and this file is needed only when the structure of the model is not just simply the main river channel, but we are also considering tributaries. For example, if you go to the Lee folders, the model Lee folders, you will see that there is no .ssf file, structure file, because the Lee model is just considering the main channel of the river Lee, while for the Sambosbrook I complicated a bit more the model, and I also consider some of the tributaries. Okay, and then there is the executable file, the .exe file, which is called branching in KN version 1012. And this is the proper interface of the model, and this is how we import the file and we run the model. So let's double click on it and open the interface. Okay, so this is the interface of the Inca model. And if we click on File, Open, we can import the files that I just described. You can do it by clicking on File, Open, or by clicking on the symbol of Open folder. So once you open the little window, you can look for the location of the files, which is basically where you copied the files from the USB memory. I did it on C, so I should have it somewhere on C. Remember, we're working with Inca and Sambosbrook. And first of all, you will not display any file because you need to change the extension down just below the file name. You will have a field called files of type, and you first of all you select the .par file. So you will display the .par file, and you just double click on it and it will open. You should display a window with some pie charts.
Is it important to open them in a set?
No.
Okay.
You can open the sequence is it's up to you. The interface is programmed in order to, if you open first the par file, then when you hit again File Open, it will show you only the .dat file, and then it will show you the second time, the third time it will show you only the observed file. So, but it's just a convention. You can open, you can start with different files.
So it just goes like one down.
Yeah. So then let's open again let's do again File Open and open the data file. If everything has gone right, if you hit the tab Time Series Data in the main window that you have opened, there is a tab that is called Time Series Data just beside the Parameters tab. You should display 3 plots. One is the rainfall, so you have in blue the total precipitation, and in red the hydrological effective rainfall. Then you have the air temperature and the soil moisture deficit for the time period that we are considering. In this case, you can see that we are considering 10 years from the 1st of January 2005 until, well, it's not exactly 10 years, no, it's slightly more than 10 years, until somewhere in June 2015. Okay, and let's open the third file, which is the observed data file. Let's start with the EA observation file, so the Environment Agency observation file. And in this case, you shouldn't display anything. Just open it. It should just say in the main window, observed data file loaded. In the main window, you have a series of rows where it just says what we are doing. And last, we open the structure file. In this case, you will have to look for the extension .ssf and select it. And that's it. You should observe a change in the little window that opened when we uploaded the parameter file. This is because before we displayed all the reaches of the river of the Salmonsbrook. Now we only display the reaches of the stream number 4. The stream number 4, I say stream number 4 because if you go to the bottom right part of this window, you will see drop down menu, and now the stream selected is number 4. If you change to number 1, which is actually the main Salmonsbrook stream, you will display 4 reaches from upstream to downstream, reach 3, 4, 5 and 6. So basically, if you browse there, you can see that there are 4 streams: stream number 1, number 2, number 3 and number 4. And number 1 has 4 reaches, number 2 has 1 reach, number 3 has 2 reaches, and number 4 has 1 reach. And this is just the structure I gave to the model because I thought it was the best structure for the things we need to do. Last thing, the pie chart. The pie charts are, at least the upper ones, represent the land use. So if you go with the mouse on one of the pie chart, for example, if you go on the red area and you click, you will see that this is the land use, and in particular this is the urban land use. The red part is the urban land use. And for example, I clicked on reach number 8, and it says that the urban land use for reach number 8 is 65%. And if you go to other streams or other reaches, you can also inquire what the land use is. For example, I'll try also stream 1, reach 3, the green area, which is arable, which is 44.89%. Okay, so that's the end of the first part.